Welcome back to our course on fluorescence spectroscopy. Today we're going to be discussing advanced topics in FRET. In the previous lecture we discussed many of the basic principles of FRET. The topics we're going to discuss today are FRET with quantum dots, FRET with distance distributions, which is by no means trivial, and the effects of dimensionality on FRET. So let's start with the quantum dots. Well, yes, they can be used as donors, or they can be used as acceptors. Here we have a quantum dot and a tetramethylrhodamine labeled acceptor, pretty far away, and the quantum dot is very much quenched by the acceptor, and the acceptor emission can be seen. It's not quite as strong, but it's clear that quantum dots can participate in FRET. Using quantum dots as FRET acceptors can be difficult. And the reason for this is because quantum dots absorb at all shorter wavelengths below their emission maxima. So if you want to use one of these particular dyes here, no matter where you excite it, it's likely going to fall into where the quantum dots are also absorbing. So you have the situation we discussed in the previous lecture, that you have to account for the directly excited acceptor emission as well as the acceptor emission that is due to resonance energy transfer. An example we'll use to go over today is FRET from quantum dots, 550 nanometer quantum dots bound to M cherry, which is going to cover the quantum dots with multiple M cherries. You can see, looking at the M cherry absorption spectra, that there's very little absorption from M cherry down in this region. So it provides an ideal opportunity to excite the quantum dots and have the quantum dots emission at a shorter wavelength than the M cherry absorption. And a theme of what we're going to be discussing is the FRET efficiency when you have multiple acceptors. We didn't deal with that in the previous lectures because it's sort of complicated. And well, this would be the equation if you had n acceptors at exactly the same distance around a quantum dot. They might not be at exactly the same distance. Well, in any event, if you have a quantum dot has a bright emission and it decreases progressively when you coat the surface with M cherry, and you can see the efficiency of transfer in a couple ways. You can see it by the M cherry emission, the acceptor emission that was excited by the donor. You can see it by the intensities, the quenching efficiency, or you can see it by the quenching lifetime. If you look at the acceptor decays, these are I like these, are quite beautiful. These are the acceptor decays for exciting this quantum dot as you're adding more and more red cherry proteins to the surface, and it keeps going down and down to shorter lifetimes, and that's because you have more acceptors. And if we drew a Jablonski diagram, we would just have multiple ways of going to the ground state. These are probably all at the same distance because the spherical dots, the quantum dots, are spherical. If you would look at the acceptor decays, they do just what we would want. Since the donor has to excite the acceptor, there's a time delay, and we see a rise time in the intensity before it drops. So that's the time from when the quantum dot is decaying, and then it excites the M cherry. And this is, as we discussed in the previous lecture, an absolute proof of an excited state reaction. Okay, we're ready on to our second topic, distance distributions. This is sort of it was very difficult when we started it. So here's what we were thinking. You have a protein in a native state, and you have a protein in a denatured state. And let's say we looked at the probability distance R at any distance from the donor to the acceptor. If it's in the denatured state, we expect that to be a wide distribution because the peptide chain is all spread out and they're not in the same conformation. But if the protein refolds into the native state, we expect it to adopt a conformation where the donor acceptors have just a single distance. Just to remind you from the earlier lecture, today we're going to be presenting data measured either in the pulse domain or by the frequency or the phase modulation method or the frequency domain. So this is just a quick reminder as to the two domains and what the data look like in the two domains. You're very familiar with the time domain. In the frequency domain, we have a logarithmic frequency scale. We measure a phase angle and we measure a modulation. The information content is basically the same, but that's how the experiments are done. So let's consider danzomelatin in two states. It can exist in these two states, and we really can't tell which state it's in from the steady state emission spectra. So we have the melatonin in either MOPS or in 80% methanol, where it likes to adopt the alpha helix. The emission intensities decay are not revealed by the emission spectra. So how do we propose to get the distance distribution? The plan for getting the distribution was to use the time resolved decays. So this is our thought experiment on how to do that. If we have a donor acceptor pair and they're at a single distance, that's going to be basically a single rate process depopulating the excited state, and the lifetime is going to get shorter, but it's not going to get multi-exponential because the acceptor, all the acceptors at the same distance away. If, in contrast, it's a denatured protein, we expect a wide distribution of donor to acceptor distances, but what we're going to measure is the intensity decays. And what the intensity decays look like is first the acceptor quenches the donor, but because there is a distribution of distances, there's also a distribution of rate constants, so the decay is going to become multi or more precisely non-exponential. There are distance distributions all over biochemistry, so I just thought I'd mention a few to you. Let's see, ion clouds can be certainly have a distance distribution. Unfolded proteins will have a distance distribution. Flexible linkers off of a protein would have a distribution of distances. So these can occur in many places, and it was our hope that the measurement of distance distributions would become more common. Now this looks crazy, but I'll try and explain it to you so you don't have to try to understand every equation. When we're doing our modeling for a distance distribution, we have to assume some shape for each peak in the distance distribution. In principle, you don't have to assume a model, but then the number of parameters is so big, like if you have a 10 nanosecond, a 10 angstrom spread, and you have all of these small increments, you wouldn't be able to resolve each of them. So we always assume some distance, some shape. So we like to use a Gaussian shape. So we have our transfer rate and we have our distance distribution. Then we have our donor decay. Now this looks pretty good because let's just like, okay, we have the donor decay and we have the rate process that is depopulating the excited state. That's wonderful, but the problem is we can't measure the intensity decay at a single distance. So it's experimentally inaccessible to us. So in order to model that, this particular intensity decay has to be integrated over the probability distribution. And that makes it look a lot more complicated, but let me just say it again. We start with Gaussian, we assume they're Gaussian distributions. We can write an intensity decay for the donor at any acceptor distance. We can't select the acceptor distance when we're measuring. So the thing we measure is going to be a sum of all of the distributions that are there. So that's represented by an integral of the probability of the distance distribution times the intensity decays at a certain distance. Not as bad as it looks. So here's a flow chart of the analysis. I like to use these once in a while. Experiments could have been in either the time domain or the frequency domain. We have our observables, which are either phase and modulation, or the other case we measure the integrated intensity decay. We then have to change that model into the type of data we're measuring. It could be time correlated photon counting data, or it could be frequency domain data. Then we do least squares analysis, and then we recover the distance distribution. So this is just an initial slide to show you that it can work, that the time resolved data does reveal distance distributions. So here we have that case we had before with a single acceptor at a single distance, and that results, it doesn't change the shape of the frequency response, which means the frequency response remains a single exponential, just shifts to higher frequency. But if the distribution is wider, then we know we have this more complex decay, which is from all of the donor to acceptor distances. So we expect a more complex frequency response, and it will not be fit by a single exponential. So the experiment is really to take this type of data for the whole range of donor to acceptor distances, and then back calculate the donor to the acceptor distance. So let's do an example case. We'll take our distance distribution, we'll calculate it for melatonin. Don't be confused by the dashed line. That's another analysis which includes diffusion between the donor and the acceptor. But if we look at the two results, in my opinion, they're really quite astounding. In methanol, where we said it had a alpha helix, the distance distribution is very narrow. And when we're just in water, we expect to have a random coil distribution. The distribution is very wide. So it satisfied our preconceived notions, and we think it's, even satisfying our preconceived notions, we still think it's correct. Now, this, I believe, is an important factor in data analysis. I mentioned before looking at the values of chi-squared in order to determine whether a fit is reasonable or not. This is a different way of looking at it, which I would call a cross-fit. We know we have two possible distributions, a narrow distribution and a wide distribution. And what I like to do is to make sure that one can exclude the other by analyzing it with the other set of parameters. So let's see which parameters we used here. It looks like these are the narrow, well, wait, I'm not going to get bogged down and try to read this from a distance with my not so good eyesight. But we tried to fit the random distribution data with a narrow distribution, and it clearly didn't fit. And vice versa, we tried to fit the narrow distribution with the data from the wide distribution, and that didn't fit. So by this cross fit, we think we've better confirmed the results, maybe even stronger than calculating the chi-square. This is an experiment that I really thought was cute. So we have a distant donor, different donor acceptor pair. Tryptophan attached to a hexapeptide glycine and a danzyl group at the end. That's one of the samples. And the other donor acceptor pair are the same donor acceptor, but they're connected by hexaproline. And we asked the question, what did we expect? And from the steady state emission spectra, it pretty much gave us what we expected. The more rigid one had less transfer and wasn't quenched as much. The more flexible one gave more transfer and the donor was quenched more. These data don't reveal what the distance distribution is. We just have interpreted these because we sort of, well, everyone knows that the proline is much more rigid than the glycine from your biochemistry classes. So let's look at the frequency domain results, which are the same ones you saw before, but now you know what the data are for. So we have the hexaproline case, and as predicted, for a single donor acceptor distance, the donor decay remains a single exponential, and it's just shifted to higher frequency, showing a reduced lifetime. In the other case, for the hexaglycine, it's more flexible. We could not fit it with the rigid model. We had to use the flexible model. And when we use the flexible model, we can recover a wide distribution for the hexaglycine linked donor acceptor pair. So that's another nice biochemical result. So I'd like to give you a couple of other examples. These are not our data. This was an experiment on aggregation of the beta amyloid peptide, and I'm sure I got a lot of this getting stuck in my brain right now. But, well, first of all, the investigators used the lifetimes instead of the FRET efficiencies. And at low concentrations of the peptide, it was thought to exist in a random state. At high concentrations, it was thought to exist in a orderly aggregated state, but in the middle, something else was going on. And this is what the investigators found. They found a single lifetime, meaning a single distance, at the low concentrations. In the middle range, they found two distributions, and they're very wide, which was consistent with their statement that the amyloid peptide was forming something like a micelle when it was at an intermediate concentration range. But when they went up to higher concentrations, it straightened out again and went only to one long lifetime. And it confirmed what they thought was going to be the structure of the amyloid peptide in different concentrations. And like I said, they use lifetimes, which is essentially the same as using distances. Here's another example from time-correlated single photon counting for the holiday junction. We might have mentioned this last time. We'll see this again when we discuss single molecule counting. But the holiday junction was labeled at different ends of the 4 strands, and it was labeled with either a fluorescein or a rhodamine. And from the time-resolved decays, first of all, we have the time-resolved decay of the donor. That's just the fluorescein alone. And then we have 2 decays with different acceptors on different branches of this holiday junction. So what it basically says is that the holiday junction does not exist in solution as a perfectly square solution. It's perfectly perpendicular geometry, but 1 is closer to 4 and 3 is closer to 2. So they learned some information about the asymmetry of the holiday junction. Here's something that's sort of tough, and I just, well, frankly, I'm not equipped to describe everything completely, but in everything we've done so far, the donors and the acceptors have somehow been linked together. So that means we were basically working in 3 dimensions. And this is the intensity decay law for a donor-acceptor pair where they are in free solution. And what is important is they have a few different versions of this. One is C0, where there's 74% quenching, but the concentration is in moles per liter or millimoles per liter. It turns out to get FRET for unlinked donor-acceptor pairs in solution, you have to have millimolar concentrations of acceptors. That's why you rarely see experiments like that done, but when you have the donors and acceptors bound to a oligonucleotide or to 2 protein subunits, that essentially makes a high concentration of the acceptor around the donor. In membranes, how are you going to define the concentration? It would be easy to describe the concentration as the number of probes per square micrometer or something like that. But what's usually used is the number of probes per lipid molecule. But notice it's a different decay law. The time dependence is different. And then what if we have donors and acceptors aligned along DNA? Well, now we're in a one-dimensional case. And again, it's another different decay law. And here we typically define the concentration as the number of probes per base or base pair in the DNA. The intensity decays in the 3 dimensions will look different if you could collect them perfectly. Of course, we have the unquenched decays, but it's easier to see in the time domain decays. The 3-dimensional solution gives you a faster decay and then a slower one, but the 1-dimensional solution starts slower and then gets faster. I cannot give you an intuitive explanation of that, but that's what's expected. And in a way, we've sort of observed that for 1-dimensional decays. But let's see, I have some data here first for a membrane bilayer, and they used a donor and an acceptor, so that's a rhodamine and a cyanide dye. And notice they use the concentrations of acceptors per lipid molecule. And as that goes up, the decay gets faster. The way this is used is sometimes you're interested in boundary lipid. That is, you have a protein that binds to the membrane, and you ask yourself the question, is there a different type of boundary lipid around that protein than there is in the bulk membrane phase? And this is detected by deviations from the random acceptor model. It has been done in a few cases. It's not easy. And I always worry that FRET across the bilayer can sort of partially look like three-dimensional FRET. Let's look at one-dimensional FRET. These are not matching cases, but I have spectra here for DNA labeled with DAPI and acridine orange. So the donor goes down, the acceptor goes up. The actual good data I have is for these particular probes. Acridine orange is the donor and Nile blue is the acceptor. And as you titrate in the Nile blue, you see it's like only one base pair per, one Nile blue per 20 base pairs. So it's not very high concentration. The frequency responses, I really wouldn't want to bet the farm on this. I think the frequency responses probably exclude the three-dimensional model, but I'm not sure they're different enough to say the one and two-dimensional models don't fit. So dimensionality is difficult to deal with. I think it'll become increasingly more important as we deal with fluorescence in nanoscale regions where they don't have 3 dimensions available to them. And that is the end of part 7B. And we'll see you for the next lecture. And thank you again for attending.